Once again, ladies and gentlemen, we're going to be talking about some more organelles. This is the last grid that we will be filling out. So our objective today is to learn structure function of the endoplasmic reticulum. Both types, lysosomes, vacuole, and the cell wall. So we're going to start out here with the endoplasmic reticulum. The endoplasmic reticulum is located in all eukaryotic cells. It is a membrane-bound organelle. As I pointed out to you yesterday, there are two different types, a smooth ER and a rough ER. The difference being that one of them has ribosomes, the rough one. So, the structure of the endoplasmic reticulum is that it's a series of tubes and sacs that may or may not have ribosomes. Okay? So, if I'm talking about a smooth ER, it's a series of tubes and sacs without ribosomes. If I'm talking about a rough endoplasmic reticulum, I'm talking about one that does have ribosomes. Now, the function, and folks, you're probably going to need to distinguish which is which, okay, which is not up there distinguished. A rough ER. A rough ER, its function is the synthesis and packaging of proteins. So, think about it. The function says here to transport proteins, all right? The rough ER has ribosomes, right? We learned yesterday that ribosomes were what made proteins. So the job of the rough ER, the function, is the synthesis and the packaging of proteins. Now, the smooth ER, which is the other one, the function of the smooth ER is that it breaks down drugs, and it also makes hormones and lipids. Okay? So you need to make sure that you distinguish between the different endoplasmic reticulums and their function, whether it's smooth or rough. Now, what would happen if we wouldn't have the ER? Well, some of these ribosomes would not have a home. The ribosomes would not have a place in the cell to attach to. Also, proteins would not have a transport system. Okay? Proteins wouldn't have a transport system. So, you can see down at the bottom there, the little shuttle train. Okay? The little shuttle train is the ER. What's it shuttling out to the cell? Okay, what's the ER shuttling out to the cell? Proteins. Okay? Proteins. This is what we're going to talk about first of all, is the lysosome. Okay, the lysosome. You might want to write under lysosome. Okay, it says lysosome on your grid. You might want to write lysosome and put in parentheses animal. And you might want to put lytic vacuole and put that in parentheses as a plant because it's called different things. Okay? It's called different things in the plant and the animal cell. you knew from your Pogel that the plant cell does not have lysosomes. All right. Now, these are in all eukaryotic cells. Okay. The structure is a vesicle that is built by the Golgi bodies. So, these lysosomes, okay, these lysosomes attach to the vesicle here that's built by the Golgi. We'll also see at times that these lysosomes will be on the vacuoles. Now, the function of the lysosome is to digest excess or worn out organelles, food particles, and engulf viruses or bacteria. So, if there is an organelle, okay, if there is an organelle, let's say the mitochondria that is old, it's wore down. The vesicle, the lysosome will come around and it will break down that mitochondria and it will see if there's any parts of it that it can recycle and reuse. Also, it helps in the digestion of food. And the final thing is, these lysosomes help fight off viruses and bacteria that the cell does not want. Okay? Helps to break down the viruses and bacteria. Now, what's going to happen if we don't have the lysosome? Okay? Cells would not be able to break down materials. So if the lysosome was not present, the cells wouldn't be able to break down materials. The vacuole. Vacuole is in all eukaryotic cells. The vacuole is large in plant cells. Vacuoles are smaller in animal cells. So there is a very, very large vacuole usually in plant cells. If you look up at the top, the blue up here, this is the vacuole. It's very, very big. If you look at the vacuole in the animal cell, look how small that vacuole is. Okay? Now, the vacuoles contain water and nutrients. Obviously, with that plant cell, the vacuole helps it maintain shape. Okay? Obviously, the vacuole helps the plant maintain shape. And we'll talk about how that happens when we discuss diffusion. That's after the cell will go into cell processes. So we'll talk about how this vacuole helps maintain the shape with the plant cell. Okay? The vacuole is a storage area. Okay? So it stores items, it helps in digestion, and it's also involved in the removal of waste. So what would happen if we didn't have a vacuole? Cells would not have a storage area for their food and water. And the plant cell would lose its shape. Have you ever seen a plant that looked like it's wilted because it didn't have enough water? Have you ever seen a plant look like that? Okay. That has to do with water in this vacuole. And we'll learn that here. Water in this vacuole. So if it wasn't there, plants would lose their shape, and we wouldn't have a storage area for food and water. The cell wall. Okay? Cell wall is in prokaryotic cells. So prokaryotes have cell walls, so do plant cells. Animal cells do not have cell walls. Okay? Prokaryotes have cell walls. Plant cells have cell walls. Animal cells do not. The function of the cell wall is to support and protect the cells. Support and protect the cells. It kind of gives the cell an outer cushion. So the cell wall gives the cells an outer cushion. If the cell wall wasn't present, okay, plant cells and bacteria would lack protection, and they would also lack the support that the cell wall provides. Okay? Would lack the protection and the support the cell wall provides. Okay? Now, we're going to look at some things. We're going to do some comparing. All right? We're going to look at some things. We're going to do some comparing. I'm going to call on some people. We'll see what's going on here. Okay? In both eukaryotes and prokaryotes, we have genetic material. Now, we know that if we are in the prokaryotes here, the genetic material is free. It is not encompassed in a nuclear membrane. Okay? So here's the nucleus. Here's the nucleus of the plant cell. But the DNA does not have a nucleus in the eukaryote. Remember, pro means no. Cell membranes. All three of them have cell membranes. All three types of these, the prokaryotes and both types of the eukaryotes, have cytoplasms. Okay? They all have ribosomes. Here's a cell wall. Bacteria and plants have a cell wall. Animals do not. Huh? What is the outermost? It's going to be like a protective coating. Okay, they call it the capsule. Now, eukaryotes. Mitochondria, right? Mitochondria, what did we say that was? Powerhouse of the cell. Vacuole. Large vacuole in plants, small vacuole in animals. Endoplasmic reticulums. They can be, Nate, rough or smooth. If it's rough, it has ribosomes. Lysosomes, or lytic vacuoles. Okay, lysosome in the plant, the lytic vacuole in the animal. Switch that. Lysosome in the animal, lytic vacuole in the plant. Okay, chloroplast. It's only in the plant, right? Chloroplast is only in the plant. Golgi in both of them. Okay? So now let's go through and look. Here we're going to go. We're going to see who knows what. We're going to see who knows what. Since Nate answered that last one, Courtney, here's yours, okay? What is that pointing to? What is that arrow pointing to? The endoplasmic reticulum. Is that a smooth one or a rough one? How do you tell? Nate, help her. How do you tell if it's smooth or rough? You just answered it. Okay. Nice and loud for everyone. If it's rough, it has ribosomes. Is that a dotted one? Yes, so that's a rough one. Okay. Jacob, what's this arrow pointing to? I think it's the little dot, not the big one. Yeah. Well, that's not a ribosome. I can understand that. Okay. This is one we talked about today. It's connected to that vacuole in the plant cell. You don't have this picture yet. Okay. Okay. Nate's going to say a lysosome. That is it. The lysosomes are connected on to those vacuoles there. Okay. What is that little squiggly stuff? Antonia? That little squiggly stuff there. See where my arrow is? What is that? Chromosome and DNA. Okay, chromosome and DNA. What is that guy? Jesse? Huh? The nucleolus. Good. Colby? What do we got here? What do we got here? That is the nuclear membrane. Excellent. I think that's pointing at that little dot. What's that little dot, Derek? A ribosome. Good. Okay. What is that guy? Brittany, what is that guy? The mitochondria. This is pointing to the white area. Reagan, that white area is? That white area is? It's a jelly-like substance. It's a jelly-like substance. We'll skip her. She wasn't here the last couple of days. She was absent. We'll take that to Nate. That arrow there, that white stuff, that big white area. What is that? Okay. Okay, cytoplasm. Okay, cytoplasm, good. What about that guy? Mia? The Golgi body, it could be Golgi apparatus, could be Golgi complex. Okay, the Golgi. What about that guy? The cell membrane, yes. What about that guy? The big circle, not those little ones, Wyatt. What is that? That is a vacuole. Awesome. Okay, good job, folks. We're pretty on with that.